Welcome Genetic Teacher Channel. Today, we are talking about colony counter, which used to count bacterial colonies, or other microorganisms, growing on nutrient agar plate, depending on seeing colonies, distinctly, and mark them, with special type of pen, on outer surface agar plate, by dividing counting colonies, into small squares, and magnify for easy count. Additionally, there are two types of colony counter, manual colony counter, and automatic colony counter, as mentioned inside slide. To use colony counter, must press on-off switch, to turn on colony counter, then, put, Petri plate containing bacterial colonies on surface glass grid, provided on colony counter, and finally, use marker pen, onto Petri plate surface where bacterial colony located, to mark colony with dot of ink, to ensure no colony missed or counted twice, as mentioned in next slide. After that, use clean and sterile Petri plates, to prevent contaminations, to avoid inference accurate counting, then, handle Petri plates, with care, to avoid any damage, that affect clarity of colony images, but be attention please. Don't overload colony counter with too many Petri plates at once, to avoid comprise accuracy. Therefore, colony counter, provide quantitative information about number and viability of bacterial samples, to understand microbial behavior, and ensure microbial product quality, to improve diagnostic capabilities, which crucial for understanding microbial growth characterization. Thanks for attention. Please like and share and subscribe Genetic Teacher videos and channel.